Dr. Hansen, the floor is all yours.
Hey, thank you very much, Andy. Thanks for that nice introduction. I really enjoyed seeing the video at the beginning of this webinar series from Balchem, because that video is very much apropos to the topic of my presentation. And what that video pointed out, I'll remind you, was that what a mother eats can affect her offspring. And that's true in late pregnancy with feeding rumen-protected choline, Reassure from Balchem. And it's also true at the very earliest stages of embryonic development, when the embryo is just a small collection of cells, hasn't really undergone a lot of differentiation. And that's true because one of the roles of methyl donors is to provide methyl groups for methylation of DNA, for DNA methylation. The methylation of DNA is critical for cell function. It plays a lot of roles in the cell, but most typically it causes specific genes or groups of genes to get silenced during specific developmental events or physiological events. I mean, to a large extent, it's DNA methylation that makes a liver cell, a mammary cell, a mammary cell. And the role of methylation of DNA in determining the function and the development of a cell begins at the very earliest stages of life. My introductory slide has a photograph on the upper right corner of a day 7 bovine blastocyst. Day 7, that's the time when the embryo in the cow undergoes its first differentiation event. Before that time, all the cells of the embryo can equally become any individual type of cell. But at the blastocyst stage, some cells begin to go along a pathway where they are destined to become placenta. The trophectoderm, those cells are called. They're in green in this slide. Whereas other cells are now destined to become the fetus itself. Those cells are called the inner cell mass. That's that small ball of cells that are orange. And I put this slide up there because the labeling is for DNA methylation. We use an antibody that recognizes methylated DNA. So the green cells, the trophectoderm cells, they have a lot of DNA methylation because they are starting to differentiate to become placenta. But the orange cells, the inner cell mass cells, they also had the opportunity to label green, but there's not much DNA methylation because those cells haven't started differentiating yet. They're going to develop into muscle, liver, heart, skin, etc., but that hasn't started. So there's very little DNA methylation in those cells. So there's really 3 take-home messages that I want to convey to you in this presentation. The first is, if we can change the pattern of methylation of DNA during the earliest stages of life, when the embryo is developing from the 1-cell stage, right after fertilization, to the blastocyst stage, that stage I just showed you, that can change how that embryo develops subsequently and even affect its phenotype after it's born. And that can persist into adult life. The video from Balchem showed how we can do that in late gestation, but even very early in pregnancy, the pattern in DNA methylation is plastic, can be modified. And we're just learning about it, but it has big implications for the characteristics of the calf. The second point I want to make is one way that we can change DNA methylation is by feeding methyl donors. And that creates the opportunity to improve function of the animal after birth, to improve its growth, to make it more fertile, or improve its capacity for milk synthesis by modifying DNA methylation very early in development. And what I want to do at the end of my talk is just give you one example of that in the cow, which is the consequences of treating the pre-implantation bovine embryo with choline and its consequences for the calf. So DNA methylation is incredibly complex, but I'm going to view it very simply. The role of DNA methylation is to change the ability of a gene to get turned on or turned off. I mean, genes are really the blueprints of the cell. They tell the cell what proteins to make and how to make those proteins. So there's two parts to every gene, the actual code that describes how to make the protein. That's kind of illustrated in this slide as target gene. And then there's another part of the gene that responds to signals that turn the gene on or turn the gene off. So during lactation, a bunch of genes get turned on. When lactation ceases, a bunch of genes get turned off. And one of the ways they get turned on or turned off is by transcription factors binding to that regulatory part of the gene, the promoter, kind of like the light switch. So when the transcription factor binds the promoter, the gene's turned on. What DNA methylation does is reduce the access of molecules that turn on genes, transcription factors, to turn on gene expression. So if the promoter region is methylated, has lots of methyl groups on it, the transcription factor is not able to access the promoter. And so instead of the gene being turned on, it's turned off. So, for any given cell, what determines whether the gene's methylated or not methylated? Well, for one thing, the signals the cell receives from outside the cell, the environment of the cell. For another thing, the diet, by controlling the amount of methyl groups available in the cell's environment. And it's important to realize methylation is not permanent. A methylated gene can get demethylated or remethylated, depending upon the physiology of the cell. What's so fascinating about DNA methylation is that it can be inherited. So if a specific gene gets methylated in a cell, that methylation can get inherited by the offspring of that cell. So you can get inheritance of turning a gene on or off. That certainly can happen by mitosis, and there's some evidence it can even happen intergenerationally. If I have DNA methylation, maybe my children will have the same pattern of DNA methylation for some genes. So DNA methylation, some people consider like the third great revolution in our understanding of biology. First, natural selection, then structure of DNA and identification of the gene, and third, epigenetics. So DNA methylation is an example of epigenetics, inherited change in the function of a gene without the gene actually changing its structure. And as I'm going to really hammer home, I hope, today, the environment of the cell can determine whether or not a specific gene is methylated or not methylated, and therefore can affect the function of that cell, and that can be inherited. So here's just a very simple example, very simple, and let's just consider muscle on the top and mammary gland on the bottom. Genetically, they're identical. Same genes are inherited in the muscle cell as in the mammary epithelial cell. But the pattern of gene expression is different because the signals that turn genes on or off is different, and because DNA methylation has silenced some genes. So in fetal life, certain genes in the future mammary cell were shut off. So for example, in the muscle, one of the major proteins produced by muscle is myoglobin. Oxygen binding protein provides oxygen so the muscles can work. The protein myoglobin gets synthesized because the gene myoglobin gets turned on. But myoglobin's not turned on in the mammary gland. Probably because the gene, down here. Probably because the gene from, I'm pointing at the slide and I just kind of realized you can't see my pointing. In the mammary gland, the myoglobin gene is turned off, probably because of DNA methylation. In contrast, another very important gene, beta-casein, that's involved in synthesis of milk protein, is turned off in the muscle, but turned on in the mammary gland. One of the regulators of that is DNA methylation. So let's look at these take-home messages. The first point I want to make is changing the environment of the embryo can change DNA methylation, and that can have effects that last for months or years and can affect the program of the postnatal animal. So let's look at that idea. The reason I think why DNA methylation is so important in the earliest stages of embryonic development is because there's massive changes in how the nucleus is methylated in early embryonic development. And then as the embryo starts to differentiate into all the different types, there's massive remethylation of specific genes in a cell-specific manner. So this is a photograph of a 2-cell embryo, 4-cell embryo, etc., that's been labeled with an antibody for DNA methylation. And the point I'm trying to make is, right after fertilization, there's still a lot of DNA methylation in the nucleus. DNA that was methylated from the sperm and from the egg. But as the embryo goes through development, it strips off most of those DNA methylation marks because it's got to turn the cells back into totally undifferentiated cells. So by the 4-cell stage, there's not much methylation. By the 8-cell stage, there's not much methylation. And then it gets put back on again. So by the blastocyst stage, like I showed you before, the trophectoderm, which is going to become the placenta, is very methylated. So if we change the environment of the embryo during this time, we can have big effects, maybe more than later in development, on the pattern of DNA methylation. And changing the environment of the embryo during this early period, from fertilization until the blastocyst is formed, has been shown many times to affect the characteristics of the offspring that were embryos during that period. So this slide just shows one experiment in the mouse where the researchers were looking at the effects of feeding either a high protein diet, 18% casein, or a low protein diet, 9% casein, on the offspring of pregnant females. So female mice were made pregnant by mating to males, like Mickey here. And then from the time of mating until the time of blastocyst formation, so only 3 days in the mouse, Mothers were fed either an 18% casein diet or a 9% casein diet. So after day 3, after the blastocysts formed, all the mothers were fed 18% casein. So this difference in protein nutrition is just for 3 days. And then the offspring were raised until 28 days of age, until weaning. Well, what did they find? They found that exposure of embryos to a high protein or low protein diet changed the phenotype of the offspring in a sex-dependent manner. And that's a big common characteristic of developmental programming. The consequence of changing the environment is many times different for a female embryo than for a male embryo. So, for example, if you look at body weight, feeding, females are less heavy than males. If you feed a low protein diet just for those 3 days, that increases the body weight of the female offspring at day 21. So just changing the environment of the embryo for that short period changed body weight 2 months later. But in the males, there's no effect. And if you look down at the bottom at blood pressure, something very sensitive to developmental programming, in both the females and males, the offspring at 21 days of life that came from embryos from mothers fed a low protein diet for 3 days had higher blood pressure, and the increase was greater for females than for males. Well, this developmental programming, change in the way an animal develops, occurs in cattle as well. The best example, the most dramatic example, is for in vitro fertilization. So we do a lot of in vitro fertilization in our lab. When you produce embryos in vitro, you have really changed their environment. Instead of developing in the oviduct and the uterus, they develop in a plastic dish. They have salt, sugars, amino acids, fatty acids, but not necessarily in the same amounts as occurs in vivo. They're missing most of the regulatory molecules that are present in vivo. They're not getting the exosomes from the mother that occur in vivo. The surface tension of plastic is different than the surface tension of the oviductal lining. So there's lots of differences in the environment. Can still get pregnancies, can still get normal calves, but occasionally development can become so abnormal as to cause an abnormal growth phenomenon called large calf syndrome. So this is a Holstein calf born in our lab from in vitro fertilization. Body weights of newborn Holsteins are about 40 kilograms. This animal weighed 98 kilograms at birth, had twice the normal body weight. Because the regulation of genes involved in growth, skeletal muscle growth, and probably skeletal growth were abnormally regulated. And here's another picture, about 1% or maybe less of the calves born by IVF have this phenomenon, probably less nowadays. Here are 2 fetuses at day 86 of pregnancy. The top one is of a typical size, weighs about 152 grams. The bottom one is twice as big, 354 grams. And you can see the liver's greatly distended, the umbilicus looks abnormal. I don't have a picture in this presentation. The placenta was about 3 times as big. So, this is an example of abnormal gene regulation occurring when that embryo was being cultured in the first 7 days of life that ended up having major effects on body size during fetal development. Okay, my second point, so I hope you agree with me, changing the environment of the embryo, changing DNA methylation can affect the adult animal. The second point I want to make is one of the ways to do that is by providing methyl donors. For in vitro produced embryos, providing methyl donors in the medium, for embryos developing in vivo, most common way, by feeding. So providing extra methyl donors through nutrition can change DNA methylation of the embryo and affect the phenotype of the offspring. Now that we've developed rumen-protected nutrients, we need to pay close attention to this idea, because now we have the opportunity that we never had before as ruminant biologists to use the diet as the way of increasing availability of specific amino acids or specific molecules like choline that play a role in DNA methylation. These two mice are genetically identical. So we have inbred lines of mice where they're all genetically identical, or 99% plus genetically identical. So these two mice are genetically identical, but they're from different mothers. So both of their mothers were fed bisphenol A, which is a reproductive toxin that blocks methylation, among other things. So you can see one of these mice incredibly large because the genes involved in regulation of body size were disrupted by the bisphenol A. You also notice this mouse is kind of a nice, pretty tan color, and this mouse is really dark brown. Again, they're both genetically identical in two different mothers. Both mothers are fed bisphenol A, which dysregulates growth, but this little brown mouse seems to be of normal body size and is of a different color. Well, why? Because of what their mothers were fed. Both their mothers were fed bisphenol A, which causes alterations in DNA methylation and abnormal growth. One of the genes that becomes demethylated is the agouti gene. Agouti causes this nice, pretty tan color. So in this mouse, the agouti gene is active, and melanin formation is such that it is this nice, pretty tan color. This other mouse was also fed bisphenol A, but it was fed a cocktail of nutrients that provide methyl donors. It was fed choline, folic acid, betaine, and vitamin 12. So its agouti gene got methylated. So it was turned off. So instead of having this nice, pretty tan color, it had this darker brownish black color because the agouti gene was turned off by the feeding. And it's not so big because the disruptive effects of bisphenol A on methylation were being blocked by all those methyl donors in the diet. So that's a pretty impressive example of how feeding methyl donors can change the phenotype of the offspring. So let's think for a second. When we talk about DNA methylation very simply as the addition of methyl groups onto the DNA, What we're really talking about is a chemical reaction where S-adenosylmethionine, a derivative of methionine, donates its methyl group to the nucleic acid choline. And that chemical reaction is mediated by DNA methyltransferase. So the methyl group is transferred from the S-adenosylmethionine to the choline, to the cysteine, providing methylcytosine. So in this experiment with the mice, they fed several molecules that would facilitate that process. So this is kind of a summary of one carbon metabolism with methyl groups indicated by the red. There's S-adenosylmethionine, has its methyl group that it donates to cytosine. Well, where does the S-adenosylmethionine get its methyl group? From methionine that was fed to that brown mouse. And methionine gets its methyl group from betaine, which was also fed to that brown mouse. And betaine can obtain its methyl groups from choline, which was fed to that dark brown mouse. And choline contains 3 methyl groups. Betaine donates one at a time to methionine, and the byproduct of that reaction, dimethylglycine, gets reacted upon by members of the folate pathway to allow those other methyl groups to be placed onto methionine. And of course, in this mouse experiment, the mice were also fed tetrahydrofolate. So this mouse, this brown mouse, got methionine, tetrahydrofolate, betaine, and choline. And that really changed how that embryo responded to being in a mother fed bisphenol A. So, you know, it is true that you can feed or provide methyl donors to an embryo, either by feeding it to its mother, or as I'll show you in a second, by culturing the embryo in the methyl donor, and change DNA methylation and the phenotype of the offspring. So the last point I want to make, because of that, there's the opportunity to regulate growth, reproduction, and lactation by changing DNA methylation of the embryo. We're just learning about this. So some changes are probably going to be good, some changes are probably going to be bad, some interventions will have no effect. But let me give you one example of what I think is a beneficial effect of providing a methyl donor to a pre-implantation bovine embryo. And that is the effects of choline on the pre-implantation embryo. So this slide has actually got a little mistake. Instead of saying, does feeding of choline program the embryo to affect the calf, which is a very good question, by the way, I should really say, does adding choline to culture medium program the embryo to affect the calf? Because that's what we actually studied. So we did a series of studies evaluating whether providing the in vitro produced embryo with choline would affect the embryo and affect the calf that embryo becomes. Okay, so again, the molecule we're looking at is choline, which is a very good methyl donor, has 3 methyl groups, and provides those methyl groups to S-adenosylmethionine through the betaine pathway. Now, of course, choline does other things. It's also involved in phosphatidylcholine synthesis, which is important for membranes. It can get acetylated to form acetylcholine, which is a cell signaling molecule. So some of these effects we see, you know, I think are probably through DNA methylation, but I can't rule out they're through some of these other pathways. So the work I'm going to tell you about it was done by Eliab Estrada Cortez. He's a research scientist from the Ministry of Agriculture in Mexico, and also working on his PhD with me at the University of Florida. And he has really pushed forward all the work that we've done with choline. And here is one of his research subjects that he first met when that embryo was a little one-cell embryo. So he did a series of experiments. I'm not going to show you all the data, where he cultured embryos produced in vitro with different amounts of choline. And this was done isotonically so that we didn't have any effect of ionic strength on the embryos. So his control embryos, like every in vitro produced embryo, is cultured without choline. Then another group of embryos, he gave 4 micromolar choline chloride into the culture medium. So 0.004 millimolar. That's an amount equivalent to the free choline in the plasma of a lactating dairy cow about 1 week postpartum. Most of the choline in the blood is not free choline, it's phosphatidylcholine. So then he had another group that received 1.3 millimolar choline chloride. And that concentration is equal to the sum of all the choline metabolites in the blood of a dairy cow at 1 week postpartum. And then another group got 1.8 millimolar. This is based on Marco Sonobi's work that feeding 60 grams of Reassure increased the total amount of choline in the blood by about 0.5 millimolar. So we did lots of studies on pre-implantation embryos. I'm just going to show you a few slides. We measured DNA methylation using an antibody to methylcytosine. And as you can see, there is an increase in the global DNA methylation caused by culturing embryos in choline chloride, with the greatest effect at 1.3 millimolar choline chloride. Actually, for some reason, a little bit less in 1.8 millimolar choline chloride. We also measured accumulation of lipid droplets in the embryo. Turns out choline really causes embryos to accumulate a lot more lipid. So in this picture, the green is neutral lipid in lipid droplets. So you can just see by looking, lots more lipid in the 1.3 millimolar and less so in the 1.8 millimolar groups. That's quantified here. Some increase, not significant, from 0.004 millimolar, but significant increase in lipid accumulation in the blastocysts. So then we did an experiment using Brahman cattle to evaluate the effects of choline added into the culture medium during in vitro fertilization and culture on pregnancy rate. There was no effect. And on characteristics of the calves. So in this experiment, we took Brahman donors, collected the oocytes by oocyte pickup, fertilized the oocytes with Brahman semen. We used a lot of sex semen, but also conventional semen. So there are more heifers than bulls. And then after fertilization, after insemination, the embryos were cultured in either a medium without choline, 387 embryos, or with 1.8 millimolar choline. And then the blastocysts were transferred into recipients. So we transferred a total of 57 embryos that were cultured without choline, with vehicle, and 43 embryos that were cultured with choline. So then we did pregnancy diagnosis, measured their birth weight, measured their weaning weight. So you can see they were put, Brahman embryo was put into a Brangus or Angus recipient. So we found some effects of the choline chloride added from day 0 to 7 of life on characteristics of the calves all the way through weaning. So in each of these next 3 graphs, the graph are the individual animals not adjusted for anything. And then the table is the least square means adjusted for sire and dam and other traits, as I'll tell you in a minute. So gestation length was actually increased by choline treatment by about 5 days, 4 to 5 days. So pregnancy was longer. Birth weight was increased by choline. And this is true even after adjusting for gestation length. There was an effect of gestation length on birth weight, longer gestation, they're heavier at birth. But even when you adjust for that 5-day difference in gestation length, the birth weight of the choline calves was larger than the birth weight of the vehicle calves. So that's about a 5 kilogram difference in the females and about a 6 kilogram difference in the males. So there's a significant effect of choline treatment, no interaction. And we're very interested in whether males respond differently than females, because that's one of the characteristics of developmental programming. And then weaning weight also differs. So there's about a 14 kilogram difference, 28-pound difference in weaning weight between the choline female calves and vehicle female calves. And then in the males, it's about a 50-pound difference. So just providing choline in those first 7 days of life resulted in 7 days more of, or resulted in 50 pounds more weaning weight. So I'm going to end, Andy. You know, I've always been interested in this equation that I learned in Animal Science 101 at the University of Illinois, where the phenotypic variation in a trait, milk yield, growth rate, whatever, is equal to the genetic variation in the trait. How do animals differ genetically? And the environmental variation. Were they raised in different environments? And, you know, I've just been so impressed at how well we in animal production have been able to improve the phenotype by modifying the genotype and the environment. You know, we are really good at identifying genetically superior individuals and propagating them. And we have spent 100 years analyzing how to change the animal's environment to optimize its phenotype. You know, here's an example of the change in genetic merit for milk yield since about 1970. But the one thing I would say is, even though we've done an outstanding job figuring out how to change the animal's environment after it's born, we haven't really thought about the fact that we can change its environment before it's born and improve the phenotype of the animal later in life. And I think that's a challenge for us, that it's going to create even more opportunity in the future. So I think I'm close to running over. So I'll just thank all the people in this slide who really were responsible for this study and for the financial support for the things I talked about today. And I'll be glad to take any questions. Thank you.
Thank you, Dr. Hansen. We've got a couple of questions that we'll get to before our next presentation. Let's start with this. So does adding choline to the diet increase choline in the follicular fluid?
You know, it's interesting. Just before the pandemic hit, we had a bunch of cows lined up where we were going to test that hypothesis directly. and then we had to stop the experiment. I don't know, but I think so. Choline is definitely present in the uterine fluid at pretty high concentration. So my guess is it's also in the follicular fluid, and that feeding choline will increase the levels. But to what extent, we still don't know yet.
Another question, was there also an increase in dystocia in the choline group in your research?
My student, Eliab Estrada, said I should point out to the listeners that none of those Brahman calves had any calving difficulties at all. So at least in this one study, there was zero dystocia. And of course, beef producers would worry about that if you had too large of a calf, that might be detrimental.
A couple more questions while we have a minute. Is there a methyl deficiency during early embryonic development?
So, that's a very good question. In our hands, when we culture embryos in vitro, yes, there's very little in the way of methyl donors in the medium. In vivo, is that the case? We don't really know. We have a grant from the USDA to allow us to test whether we can see the same kind of effects by feeding rumen-protected choline in the first 7 days of embryonic life as we see in this really artificial system of the in vitro produced embryo. So we'll try to find out the answer to that question.
And that may be a good segue into this, which is probably going to be our last question for you. What are the critical stages during that embryo fetal development where DNA methylation is the most important?
You can find examples in the literature throughout the entire range of gestation, from even before fertilization, the oocyte, the egg in the female, can get modified to affect the offspring, all the way through the dry period, the last few weeks of gestation. So, we don't really know enough about which period is most amenable to manipulation.
And I would just say we had quite a lot of questions come in here literally in the last minute. And so if we didn't get a chance to answer your question, our presenters and folks from Balchem, somebody will get back with you after the presentation to answer your question. But appreciate all of these questions. And just a reminder to all of our attendees, you can, what's that, Dr. Hansen?
So I kept going.
Yeah, well, it's good stuff, right? So appreciate an in-depth presentation, and thanks to Dr. Peter Hansen from the University of Florida for a really insightful discussion, and look forward to catching up with you again at future presentations. Before we move on to our next presenter in this virtual mini symposium, I want to thank Balchem Animal Nutrition and Health again for sponsoring today's sessions.